Our next speaker, Dr. Stephanie Slinsky, is the Associate Director for the Yuma Center for Excellence in Desert Agriculture, YCEDA. She is a plant pathologist by training, with a broad research background in fungal and bacterial diseases of vegetable crops, conifers, and citrus. In her current role, Dr. Slinsky has experience working with researchers and with the agricultural industry to develop solutions to the many pressing production needs of our region.
Well, hello everyone. Thanks for joining us today. I'm the Associate Director of Research at the Yuma Center of Excellence for Desert Agriculture. I'm also a plant pathologist. At the Center of Excellence, we are working to support desert soil health research and with the goal of helping growers make good management decisions. I'm going to talk to you generally about soil-borne plant pathogens and the influence of soil health and disease, and I'll also discuss some methods for suppressing disease while supporting soil health. Okay. Soil-borne pathogens come from all plant pathogen groups, including nematodes, bacteria, fungi, viruses, and oomycetes. And here's just some examples of that. I want to point out that this, under fungi, that's the fruiting body of the Sclerotinia sclerotiorum fungus. These pathogens may develop similar symptoms because they attack the root or the crown of the plant. And here's some symptoms of, some pictures of, sorry, my computer's freezing a little bit. Here's some symptoms of 4 lettuce diseases that look very similar. Complicating the situation, these diseases can occur in the same location. For example, in California, growers are seeing Pythium and Fusarium wilt occurring in the same field. It's possible that without the Pythium infection, the Fusarium wilt would not become a problem, as Fusarium has been a resident of California soils for decades without, often without causing significant crop loss. One way to differentiate the diseases is to examine the crown and the root system. These pathogens produce pretty distinct symptoms. You can see that Fusarium wilt and verticillium wilt, those are both vascular pathogens. And so you have to slice that root open to see those symptoms in the vascular system. And you can differentiate these pretty easily. The Fusarium wilt generally has the brownish or reddish symptoms, where the verticillium is more a bluish or blackish symptom. With lettuce drop, you see the sclerotinia in the crown. You also see some signs of the disease there with the mycelium and sclerotia, but that's distinct from Fusarium and verticillium because you have that rotting of the crown. And then another example here is Pythium wilt, and that gets into those fibrous roots and rots the root from the outside. These pathogens also produce different survival structures. These are durable structures that can protect against desiccation, UV, and other environmental stresses. Some examples are the chlamydospores of Fusarium, sclerotia, verticillium, and sclerotinia, oospores of Pythium. These pathogens can survive using these survival structures for years in the soil. And there's also other mechanisms of survival. Other mechanisms are something like alternative hosts, like weeds or in crop debris. These act as a reservoir of these pathogens. Some of the pathogens like, Fusarium, Sclerotinia, they will survive in these types of materials. And there's also some pathogens that this is their primary survival mechanisms, like with Rhizoctonia, that's here, that it primarily survives as mycelium in crop debris or in other hosts. And also viruses can survive in weed species or in other vectors. Bacteria, and here's some root knot nematode infected roots. So there's many mechanisms for surviving in the soil. If you sample the soil and analyze the microbes found in that sample, you might find many pathogens present. However, disease may not occur, even if the presence of a highly susceptible, even in the presence of a highly susceptible plant. Here is a little video of Pythium sporangium germinating. It releases these motile zoospores that are attracted to the sugars and the amino acids that are exuded by the plant roots. They swim towards the roots and lose their motility, germinate and infect the plants if nothing stops it, if it has the opportunity to reach those roots. Here's an unanswered question of when does disease occur. Pythium insinulatum has been found in Arizona soils for decades, and it's been isolated from lettuce roots that did not show symptoms of a disease. But this pathogen causes severe disease in California on romaine lettuce. We don't have a problem with this in Arizona. I don't have an answer to why we don't see this disease of the Pythium wilt in Arizona, but I can explain the factors that are necessary for disease to occur. Historically, we've used the disease triangle to describe the factors necessary for disease to occur. I like the Venn diagram. I think it really shows you how you need those overlapping factors to have disease. This works if you don't have a vector, if the disease doesn't need a vector to infect a plant. Only when these three factors, the susceptible host, conducive environment, and pathogen occur, will disease occur. A corner of the disease triangle that is quite obvious is the pathogen, and this, I gave you some examples of this in previous slides. A susceptible host is another corner of the disease triangle. This could mean a host that does not have any genetic resistance against the pathogen. This could mean that the host could not put energy towards defense. It could mean a host that was not primed to resist a pathogen attack. Could mean a host that did not receive a pesticide treatment that prevents infection. It means the host is available for colonization. A conducive environment is the final corner of the disease triangle. This is an environment that promotes disease by favoring the pathogen. This could be the climate, such as temperature. An example of this is Fusarium multif lettuce will develop in Yuma on lettuce planted in early and mid-September when the temperatures are very high. The pathogen likes these warm soils. This is also a time of year that really promotes that plant stress in the lettuce, because the lettuce prefers to grow in much lower temperatures. So, we might be seeing, 90, 100, 110 degree weather in September, while the lettuce wants to grow in 70 degree weather. So that makes the host more susceptible. Soil quality is another factor that could influence both the environment and the host susceptibility. Soil health is an important part of effective plant disease management, effective long-term disease management. Soil health and plant disease go together. Often we see disease as an indicator of problems with the soil. This can be caused by plant stress or could be caused by high inoculant levels in the soil. It's an indicator of a less diverse microbial community because an active and diverse microbial community will not support high populations of a single microorganisms. And to add to that, you also need to have a significant biomass of that microbial community. Plant stress does not always cause higher levels of disease. With some diseases like downy mildews, less disease may occur in stress tissues, but I'm focusing on soil-borne diseases here. I'm glad that I don't need to go into soil health since that's been well covered in previous talks and I'm not an expert in this area. I do, however, want to remind you that there's 3 general factors that are part of that come together to form healthy soils. That's the physical, chemical, and biological aspects of soil health. I want to bring these, remind you of these so I can introduce you to some factors that contribute to plant stress. So here's some examples of those physical, chemical, and biological stressors. It's important to understand that those physical and chemical stressors will also have a negative effect on the biological aspect of soil health. So they affect those soil microbes and the other biological elements to the soil. We'd like to have healthy soils for effective long-term soil-borne plant disease management. We want to have the physical and chemical factors to promote plant growth and reduce stress. We want a stable, active, resilient, and diverse microbial community. Of course, I mentioned there's other aspects of the biological community. In the next slides, I'm going to talk about the microbial communities. So you know that healthy soils reduce plant stress. Reduced stress also allows the plant to focus more energy on defense against pathogens. Physical and chemical factors are also important for suppressing pathogens separately from plant interactions. There's some studies that have shown, for example, that changing the soil clay composition or pH has influenced pathogen populations. Microbial communities in the soil can significantly reduce disease pressure. Some microbes can help build up the plant defenses by stimulating the plant to produce defense-related chemicals or physical defenses against the pathogen. But competition in the soil environment is a much more general method to suppress disease. This could be by occupying the same niche as the plant pathogen, especially in the rhizosphere, which is that area around the roots influenced by the sugars and amino acids that are released by the roots. Some microbes will colonize the surface of the roots or will grow into the roots without causing disease. This is a very effective method of excluding pathogens from the root. There are soils that are recognized as suppressive soils. These are soils where the pathogen may be in the soil, but causes little to no disease. It is possible to have symptoms. Usually the symptoms are a lot less severe than if the soils were not suppressive. The soils could help prevent pathogens from establishing in the soil. One of the characteristics of suppressive soils is that they have a diverse microbial community, which is an indicator of soil health. And usually no one organism is responsible for that suppression. However, there are soils that are suppressive to specific pathogens. And this is usually from the effect of a specific organism or group. As an example, there's some soils that have been identified as suppressive to fusarium. And there's several organisms that have been found to be responsible for this suppression. One organism that's been found to be effective is a non-pathogenic fusarium species, or generally non-pathogenic fusarium species. Fusarium oxysporum is ubiquitous in the soil, and many of the Fusarium oxysporums that are found in the soil are not pathogenic on any plant species. And these can outcompete pathogenic fusariums because they are occupying that same niche. FO47 is a Fusarium oxyspermum isolate that was originally collected from a melon field that was suppressive to Fusarium wilt of melon. This is from a field in France. It was collected in the late 80s. It's been found to be effective not just against Fusarium wilt of melon, but against other fusariums and other fungal species like Verticillium dahlia, which is a common, it causes verticillium wilt. And this FO47 has also been found to be more effective when combined with other organisms like specific species of pseudomonads. And there are other bacterial species that can also help compete in the soil environment. And it's often because they produce chemicals that will suppress pathogens, like antibiotics. So streptomyces make streptomycin. So there's different chemicals that are produced by these bacterial species in the soil. Unfortunately, FO47, there's no products that are made. There were 2 products from Europe that were made with FO47, but they're not available. So it doesn't seem like, even though this research is ongoing, I saw some research papers from this year that report on studies with FO47. It doesn't seem like there's much of a move towards developing a product. But it's good that research is continuing, and there's other species, there's other organisms that have been found to be effective against Fusarium. There's a Colomonas isolate that was isolated by Johan Lavau's lab from UC Davis that was shown to suppress Fusarium wilt of tomato. These are studies that are pretty early on, so they are not being performed at field scale at this moment, but there are opportunities to develop products that could help develop suppressive soils or less disease receptive soils. But a healthy soil is the starting point to all of this. When you decide to start developing suppressive soils, you may decide to use a soil inoculant. There are many inoculants on the market right now, but the inoculants alone won't build a stable, active, resilient, and diverse microbial community because, as you know, the soil system is very complex. The microbial community is complex, and it's not possible to create a formulation with the level of diversity that the natural soil environment has. But, and formulations need to be adapted to your environment, and the soils need to sustain the microbe in the formulation over long periods of time, including those host-free periods. But, you know, as you understand from the previous slide, there's a role for inoculants that can provide, if they can provide the appropriate competitive microorganisms. And there can be a role for some of these products along with other methods to build soil health and support the native microbial populations. Commercial inoculants are labeled as having various modes of action. I discussed some of these modes of action in previous slides. These inoculants usually contain microbes from a limited group of genera, and this is because these are groups that have been identified, which means they can be grown in a lab or grown in a fermenter. They've been evaluated for specific modes of action. They can be formulated into a product, so they have a shelf life, so they can actually be used in agriculture. And the genera that I listed are just you know, maybe half of the common genera that are found in a lot of these products. It's difficult and expensive to evaluate products on multiple crops and growing regions, so efficacy data may not be extensive for your specific growing conditions in your crop. So you should carefully consider any product you decide to add to your soils. Consider your goals for your soil and use products and conditions that can sustain the microbial populations. There are some other methods to suppress disease and build soil microbial communities, including biofumigation. Biofumigation is the use of a brassica green manure that releases isothiocyanates when the debris is incorporated into the soil and irrigated. This is a chemical similar to the active agent of the fumigant metam sodium. And the addition of the green manure helps to build soil organic matter and build the microbial biomass in the soil. The most consistent success with this method is against nematodes. And this may be because nematode suppression does not rely completely on high levels of isothiocyanates. There are other chemicals in the green manure that contribute to the suppression along with the isothiocyanates. Biofumigation has been used to suppress many pests and pathogen groups, including nematodes, weeds, insects, fungi, oomycetes, and bacteria, but with mixed success. Timing is really important, both for when you have the highest level of the chemical in the plant and when the target is most susceptible. So you really have to time those so they occur at the same time. The method of incorporation is really important. You have to pulverize that plant material and immediately irrigate. The soil conditions have to be managed to maximize the production of these chemicals and also to minimize the volatilization of the chemical. It's a promising method of suppressing pathogens, but there's not yet consistent efficacy under field conditions. This is something that I've done a little bit of work with recently, and also Alex Hu from the University of Arizona in Tucson has been doing some work. The background of this slide is some mustard that we planted this spring, and we did a pilot study to look at suppression of fusarium wilt of lettuce. Another method of suppressing pathogens is by soil biosolarization or anaerobic soil disinfestation. The difference between these two methods is only that biosolarization is conducted during periods of high heat, which we have lots of in Arizona, whereas anaerobic soil disinfestation does not have the heat to get that solarization effect. ASD and SBS are, like I just said, they're similar to solarization, but a carbon source is applied at a rate of 4 to 9 tons per acre, and the soil remains saturated for the duration of the treatment. You put this plastic on top, you saturate the soil, you have the carbon in the soil, and that induces anaerobic bacterial growth. It's hypothesized that the anaerobic bacteria produce chemicals that are antagonistic to the pathogens, but the mechanism may also be through antagonism, through niche competition. This method may suppress pathogens longer than solarization alone by more effectively reducing the pathogen populations. But it also might be because with the addition of the soil organic matter and the stimulation of the microbial populations, you're maintaining suppression of the pathogens for longer than solarization. Solarization can have a detrimental effect on microbial populations because you're just heating up that soil, you're killing a lot of those microbes in the soil. It's similar to fumigation. Anaerobic soil disinfestation is currently being used in California to treat more than 2,000 acres of organic strawberries. It does a really good job suppressing soil-borne pathogens. mainly verticillium, and it also suppresses weeds. The background picture of this slide is an experimental field in California looking at anaerobic soil disinfestation. I visited this field last March. ASD and SBS have been extensively studied in Florida and in Tennessee, and we were starting some work in Yuma to evaluate SBS using against Fusarium wilt of lettuce. If growers are going to use this treatment, we need to develop economical methods to do this, and we need to see long-term effects. There's been studies combining biofumigation and anaerobic soil disinfestation using green manures or using mustard seed meal, but more information is needed to determine if this is a viable option and if it adds any benefit to the ASD treatment. Mustard meal may not be economically viable since it's a pretty expensive product. Here's some very basic conclusions. Even more succinct is to say that healthy soils mean healthy plants and less disease. This sounds really simple, but of course it's not, especially when we don't know exactly how to quantify a healthy soil in desert agriculture. So thank you, and I just want to thank Steve Kowicki, Robert Wick, and Tom Gordon, who gave me some great photos to use.